We don't have to install anything on our system. We can have a very rich resources and but there is also some drawbacks. What drawback is, for example, if we don't have the very good utility connection and any time if our connection lost, we have to restart the system and that's once again. And the other thing is when we are doing online, we install something and when our session is expired, every installation is expired. We have to reinstall everything. So let's see first what is the online possibility. We have the full app from Google. Full app. Google. So here we can do online, for example, facility. Let's say, for example, new notebook and when we have the new notebooks, we have to select some features and we can start our AI programming. And if our library is not installed there, we can install ourselves. This is just to tell like what type of feature are there, like we can start generating the code here. Just take a simple example, print. Hello world. So it means everything is there. I didn't install the Python. And even if I need the high resources, I can choose another processor, TPUs. But these facilities, we have to have a very good internet connections. And the other thing is whenever we close our session, our all installation, all the packages are lost. So mostly we prefer for offline versions. For offline versions, we have to see like what are the top possibilities around the world. I have, for example, we say there are three top possibilities as per my knowledge. Maybe there are some other around the world. One is we can use the PyCharm. It's a part of Anaconda. We can try the VS Code that's from the Microsoft, or we can try the Jupyter notebooks. All they are free. Some of come with rich libraries, and for some we have to install the libraries. So let's have a look for example. I have already downloaded, for example, AI programming. So here I have the Python 3.30. We must have to remember when we go for AI programming on the back end, we need an interpreter that is a Python interpreter. We can download from python.org. And for every system, either it's a, let's say, VS Code, either it's a PyCharm, either it's a Jupyter notebook, all of them used on the backend Python. Python is a language that is available. We can download it. We want to download it separately. Let's say, for example, we say so here it's available free. We can install it, download it, and install it. But for Anaconda, it's already part of the Anaconda. Anaconda suite have many other utilities, and also Python is already integrated there, so we don't have to download it separately. But if someone wants, he can do it. Let's see, for example, I have downloaded already Anaconda. This is the latest version that I got yesterday from the internet. So we have to go install it. So at this stage, of course, my system is 64-bit. It's I have downloaded for 64 bits. And next, here is agreements what I have to do. Of course, it's a license agreements for what purpose I am going to use. And this at this stage, it asks two things. One, it asks just me, for example, for the user that is logged in, it's this installation will be valid for that user, or I want to have all users who use this machine. normally I will go for just for me. So here if I have to remove a little bit, maybe some another stage I have to know where it's installed. Okay. So like this is like my user and this Usama is my computer name and here is the Anaconda 3. This is the Anaconda version number 3 and this is the installation path. Maybe later on somewhere we have to know where it's ready installed. So then just I go for next. Here we have to remember a little bit few things. Here it says add Anaconda to my environment variables. On Windows system, whenever we install something that must be a part of system variables. So we will say yes, add Anaconda 3 to my environmental variable. And I will say also register Anaconda as my default pattern. Then you can claim the cache. It's not mandatory others. Let's say install. So it's really very fast. If you have a good machine, it's will install in a few seconds. And then we can start it. We can see if we want to see the details here, what it's installing, which package is on the behind is going to install. Other we can close it and it will install in a few minutes.
Sir, here is a question, either it is an Anaconda Mini or it is a standard Anaconda.
We use the standard Anaconda because it has a rich feature as compared to the mini one. Just we have to wait. So there was some question we can take it from the machine.
My question is, the cargo bit of things from here.
Question answer.
Question answer.
Thank you.
Sir, the questions for the job, can you answer the question? I am the more type.
Yes, we can answer.
I have not, Ms. Sabha Kamal is asking, I have not marked the checkbox for setting the environmental variables as it was giving a warning. Please guide another way to set up the variables through command prompt.
So normally it's better always, we should have a one conda environment on our system. And one suit, we can say, and we can add up system variable, for example, we can say system variable. Over there, we can add our environment variable, and we have to tell where actually our installation main folder is. Initially, like I show you, it's go for single user, it's the users and user name, and then it's going in the machine. And then we have to give the path over here. We have to say new, and then we have to say, for example, it's in the C drive, and it's users and whatever the name is, for example, Osama. And then on the 35, we can add in this. If we don't check the checkbox during the installation, it do automatically.
I guess we should take this all the environment settings as default. It will work as it does.
Yes.
If we not check any box and state it as default, then it will be quite fine because we are only having, we need only having the pandas, we need to have numpy and scikit-learn. So then if we just go with the settings with the default, it will be working fine. Yeah, so let's make it very simple. So I guess they should just click on the next and next and next. So it will be easy for them to just install the Anacondas.
So our installation is complete, and let's have a look how we can program it. Here it says like Anaconda and Jupyter. So we can launch the Anaconda navigators. So do remember Anaconda navigator is it needs more resources. So if your machine is not so good, then it's better to install the Python interpreter and maybe Jupyter directly.
Yeah, because huge memory of the RAM is being utilized by the Anaconda when you install. It is better to have a system of minimum 8 GB RAM so that you can use the other specification. You can use the other applications while using the Anaconda. Somehow, like once you are installed with the Anaconda, then you are fine to go. Then you need to install, and then you need to launch the specific program that you need to work on. Like basically, do you guys know what exactly the Anaconda is? No. That as the sir has said before, Anaconda is basically it's like a platform that will provide you an environment that is actually this soon. That will provide you the multiple environment. Like it contains the R programming language, it contains the Jupyter through which you can use the Python, it contains the PyCharm. In fact, you can use the Python in it. And again, it has the multiple applications built in it. It provides you an environment so that you can use those in the tools or you can use those packages feasibly. Another way we are actually heading towards the installations for the Jupyter Notebook because we are actually targeting the Jupyter Notebook, right? So there are two ways actually to install to have the Jupyter Notebook in your Windows. You can even install the Jupyter Notebook while being through your command prompt. Like you can just give a command to your command prompt. You know that you have the CMD, the command prompt in your Windows, right? You can even just give a command. You can just type the install Jupyter Notebook. It can easily download the Jupyter Notebook. But actually, what we will be facing, what exactly we're gonna struggle in that will face, that will give us like you need to install multiple packages. Like there are some packages that actually that Anaconda gives you. But if you install the Jupyter Notebook and through the Anaconda, it will actually gives you the built-in tools that will you can use in it. You can just install it. You can just call that tool and just you need to work on it. Okay. But if you download the standalone Jupyter Notebook, you need to install every basic tool. Like that is even you want to set up your directory, you need to install that tool even. Okay, so that will be more hardest thing, or that will actually frustrate you. So that is actually I believe like that platform is very much basic for you, and this is actually the first step you're gonna have in the programming language. Most of you maybe that most of you are using the Python or the Jupyter Notebook for the very first time. So let us make it very friendly. Let us make it very useful and practical to use, easy to use. So that is why we are actually targeting the Anaconda. We are actually targeting the Jupyter Notebook through the Anaconda. While using the Jupyter Notebook through Anaconda, you just need to call that tool, and then you can just program and just code your.
So as matter of fact, like with the Anaconda, we have a lot of features. If we look at, for example, PyCharm, Anaconda Navigator, here, what is, which you can see here, installing, it's not installed by default. And if we go down, for example, Jupyter Notebook is the package inside the Anaconda source, so we don't have to install it. And much other libraries, for example, Panda, NumPy, and other TensorFlow libraries, it's as part of its. Let's see, for example, if we go to the environment, you can see it's not installed only the Python, it's also installed and other packages. Total are almost 525 packages are already installed inside. So, and then other thing is like we have the environment variable here. Later on, when you and all of us will get more experience, then we will see we have some special feature in Anaconda we can install, and at the same time we can have a multiple environments. Why? Sometime really, if you go in the industry and you will see some packages what is available in the market, that is the first special version of Python. For example, they will say you can, it will run perfectly on Python 3.10, but not in 3.30. So here it will provide us that we can create the and other environments. But here at this moment, we don't need, and we will go, for example, to home and we can launch here. For example, we say Jupyter Notebook and we'll say launch. It will start the Jupyter Notebooks. And over there, we can start our coding, like how we can start it.
So once the Jupyter Notebook is installed and launched in your system, it will redirect you towards the web browser. Whatever the default web browser you are using, whether it is the Firefox, whether it is the Google Chrome, it will redirect towards that browser. Okay, and that actually does not mean that it is the Jupyter Notebook you're gonna use is dependent upon your internet. So this is the offline. You cannot, if you want to install any other package, you need to have your good internet speed. Need to have the internet connection to get that package installed. But most of them we're gonna use is actually is already present there.
So in simple, like this Jupyter Notebook runs in the browser, but it does not mean it's over the internet. It's only need the browsers. Over here, we can come when we can see here our working directories here, or we can say, for example, new, and we can start the new notebook here. And with the new notebook, we can rename it, for example, what pattern kernel default is, we leave it pattern, high pattern kernel selected. And I can rename it, for example, demo. And over here, I can start program. And for example, we did just only print hello world, like for example.
So every one of you is able to install the Anaconda?
So then we have to look, for example, we can say run, it will run it, we see that's the output is there. And in run, we have many other things. For example, one thing mostly we use, we say the show line number. Sometimes when some error comes, we can see which line is exactly is. And if I am here and I don't want to execute this above code, then I can say, for example, run all cells, run the selected cell and all the selected cell below, whatever I want. And over here, if any reason my kernel is crashed, I can restart my kernel here and can say restart the kernel and run it. So this is just hello world example. We can do a lot of other things. For example, let me show you one nice demo. I have one demo here in my downloads. Programming for AI, I have a demo. For example, this is a solution to question 1. So look here, what we have studied there, KNN, KVM clustering, and over there by all we can use these libraries, these pandas, NumPy, MATLAB, sklearn. These all library is already part of the Anaconda tool. So I will say, for example, when I come here, I will say, let's say, run all cells. I can run cell by cell. I can run all cells. Like here we can go up, and we will see the star shows it's working behind it. This is done, and now we have he is on this stage. What it is? We have some data set that California housings, and over there we have some special feature: income, house age, house room, bed, occupation, all this, and then we have can do some type of clustering over there. We can apply elbow methods, how many cluster we need, we say like 5 is the best one, and so on. So in similar way, for example, when we are working for medicine, of course, we will have some library that supports medical imaging, some other stuff. Some specialized libraries are there, we can install there. So this is the one way. So let's summarize all the things together. We told initially we have 2 possibilities. One possibility is we can do programming for AI using the online availabilities. One availability, the one of the best availability is called the Colab. On the Colab, we go, for example, new notebook, and we can program whatever we want. So the first thing I want to mention here, it needs very good internet connection. And if I need some installation, I can install the package, but whenever I will close this session, everything will be lost. Because these free versions, the third-party companies don't allow you to install a lot of things on their systems, and maybe you don't complete. And so the best way is to have the offline versions, and I show you for offline versions. The best possibility, if you don't have the good resources, mean not a very good laptop, you can install only the pattern. And you can install maybe the VS Code and maybe the notebook. And PyCharm is also can be installed. So the slight difference between PyCharm, notebook, PyCharm provide us a lot of feature. For example, as I told, like we can have a multiple environments. I can have a same time pattern 3.10, pattern 3.13, whatever I want, what is supported inside. But and then I have to have one, maybe the Jupyter notebook, maybe the VS Code. VS Code is a lightweight. It provides us also the other features like debugging. We can do the debugging, but Notebook Repeater Notebook still has some feature newly introduced, but it's not so rich in debuggings. So, and PyCharm is really very good in the debuggings. So I show you, like we can download the Anaconda. Of course, it's available free of source. And then we can install, like I told you how we can install it. And then we have this base environment by default. All the packages are installed there. And we can launch our notebook and we can start a program. So this is a small from my side. If any of you have some question, we are here to answer it. Ma'am, maybe you want to say something?
I have questions. Questions for
some Anaconda the coaches already installed है कुछ तो इसके ऊपर तो लिखा हुआ है launch जिनको पे install लिखा हुआ proper जाके install करना पड़ेगा.
Of course, we have to say we should have internet connection and we say install, it will install automatically.
अगर download का option है सर.
Anyway, it install, it will download and then start installing it.
Maybe we can take the question answers.
Yeah, maybe short. Yes.
Most of them I have answered from a mobile. Yeah, can we use the Anaconda on the Android? No, we cannot use the Anaconda on the Android. We need to have the laptop or workstation on the computer
Yes.
in front of us. Yeah, you can use it. Which Anaconda we have to install distribution installer or Miniconda. That's the distribution you need to install. The main Anaconda can navigator, not the Miniconda, not the Conda, Anaconda, the main one.
The standard one.
Do we need to add any environment variables? I guess you should go with the default settings. And then the most of the packages environments are already installed in that Anaconda. So you don't need to go with the specific one. Sure. After opening the Google Colab, what should we have to do? Please share through the screenshot. So can you go with?
Yeah, we have to go, for example, new notebook. For example, let's say here. We have Colab.google. This is our. Then we have to say new notebook. Open the Colab if I already have the notebooks. I will say the new notebook. And here I can start showing whatever I want.
We can install some other packages like and pandas and done faster.
Yes, of course.
Once they're done with the installations.
For Jupyter notebooks, initial pattern is there. Of course, when we want to use the other libraries, we have to install them. And that's one of the feature of the Anaconda. That's a part of the suite. The standard libraries are there. We don't have to install it, but it's a little bit resource hungry, or it's need more resources. Must have a good machine. So it's a choice of everyone how he or she wants.
Basically, the Python is used by the everyone, like a person or the individual from every domain. Even the Python is being used by the linguistics people. Even the Python is being used by the statistics, the bioinformatics people, the engineers, the mathematicians. So Python is a big, or you can say the umbrella, and you can use it up to your work, up to your interest, and you can install the tools or the packages, whatever you need for your work, basically. Okay, so there are some of the packages, there are some of the tools that are universally used, that can be used by everyone. Like you need to set up the directory. You know, guys, what is actually the directory is? Yeah, that's the folder. Anaconda is basically installed when you were installing your Anaconda, it gives you a certain path like C users and your username, and then it creates a folder that was the Anaconda 3, and then it installed all of the files that were quite relevant to the Anaconda that were being installed into that specific folder. Okay, so if you check that where exactly I am present, so it will tell you the location where you have installed the Anaconda. So you need to drag it, you need to take it towards your file, but where are they present? Whether they are present on the desktop, whether they are present on the D drive or the E drive or any folder where it is. So you need to set up a directory. Directory is basically a folder, like you contains a folder, like in the desktop you have multiple folders, you have multiple files. So you need to take your Anaconda to that folder, to that directory. While setting up your directory while guiding your Anaconda or the Jupyter notebook towards that folder, you again need to have a package to be there in the Anaconda, to be there in the file. So that is actually the OS. Okay. And while reading your file, obviously we are going for some data analysis. We are going for some machine learning, as you have studied some classifiers or the some supervised machine learning models, and we are going to implement them actually in the Python, right? And on which file you're going to implement it? There must be some data set. You need to read that file in the Python. To read that file in the Python, तो इस पे हम उनको जल्दी से कमांड बता हमें कमांड लाइए जी
Not taken, so not.
नहीं we need to have the numpy जी numpy pandas and scikit-learn
Yes.
तो वो चीजें हम बता हमने तो वो what we can do is maybe we can do it after Dr. Chen's time.
Then we
Is it
can
okay?
do it after change no issue. And also
Okay, sir, आप please stop करें
yes.
Okay, Professor Chen, can you hear me? No.
Oh, yes, I can hear you. Can you hear me?
Okay.
So,
ये mic.
Okay, Professor Chen, kindly share your slides.
okay, I'm sharing my slide. Can you see my slide now?
Dr. Chen, I request you to please begin your lecture, number 5, that is on unsupervised machine learning methods. This is lecture number 5 of a 19-day long course on applied biomedical AI, which is being organized by COMSTECH-NINGBO University of China. Professor Dr. Chen is Deputy Director at State Key Lab of Chemical Oicogenomics. And with this, the floor is yours, sir. Kindly begin your lecture.
Oh, okay. So, in today's lecture, of course, we are studying on supervised machine learning methods. And afterwards, we are going to have a.
We cannot hear you.
Let me see. Can you hear me? That should be no problem. Can you hear me? Can you hear me?
Yes, we can hear you, sir.
So we can continue. So after today's lecture, we are going to have the practical session. Dr. Wang, we're going to get you through the session. Please bear in mind that he is not good at English, so he may actually utilize some internet resource in order to be to be able to actually go through the practical session more smoothly. So we give the full understanding of whatever he prefers to do. Okay, so in today's lecture, we focus on unsupervised learning methods. So this is in contrast to what we have learned so far, the classification method or so-called supervised method. So in supervised method, if you recall, essentially we have a data set, we know the right answers. We want the AI to be able to repeat our success by his own learning, his or her some kind of learning process or by its learning process. But there's another situation that we do not have the knowledge, so we cannot teach the AI to get a correct answer. Instead, we seek AI to analyze the data on its own and then provide us with some hints about the characteristics, about the patterns, even about the essentially the intrinsic label of the data. So this is called unsupervised learning method. Actually, in our deep learning age, particularly in the big language, large language models, so the most important step is this unsupervised learning, because the more data we got, the more vast the data inside, the less capability we actually get all the labels of the data. But on the other hand, we have so much unlabeled data. So we just handle the data to the AI, and maybe AI can figure something out. So this is what we are going to learn starting from today. So in an unsupervised learning process, essentially the AI tries to distinguish the data, each individual data, based on the comparison with the other data. So that's a comparison among the data. So the comparison means that some data may be look like each other, may be similar to each other, some may be more distant away from each other, right? So from this sense, you can already sense that there must be a need to define what constitutes two samples that are being close. What constitutes a situation where the data actually are far apart. So there's a need to define distance and similarity. This is not a trivial task, as you will find out slightly later on. So after we get over with the distance, how we define the distance, we can look into three specific methods: hierarchical clustering, k-means clustering, and fuzzy c-means. Okay. First, regarding to the distance, of course, we may think this is a trivial question, right? So if there's two samples at each at a particular position, like this figure illustrates. Somehow there's something wrong. Okay. Okay, so obviously the distance must be the just the Euclidean distance, right, between these 2 points. So that's what we commonly define as a distance. But in data science, actually, this Euclidean type of distance may not be the best definition of distance. There are some other issues we have to consider in terms of the definition of the distance. So apart from Euclidean distance, there's also this so-called Manhattan distance. So and then there's some other definition like a correlation coefficient, correlation measure of the similarity of the data or the distance of the data. So the Manhattan distance is the purple one. So essentially, it is the distance along the horizontal line and also the distance along the vertical line. And that means you don't go from one point to the other point in a straight manner, but rather you go horizontally and then vertically. And then the correlation means that we regard the 2 points as the points of a vector. So a vector, we know that has 2 components. So it is illustrated in this slight yellow or yellow-brown color, the dashed line. So each line has essentially has x and y 2 components, and those components define the vector, right? So there's this kind of vector correlation type of distance, which we will look into the details later on. But first, we want to look at why for instance, the Manhattan distance is introduced. Essentially, we know that in real situation, sometimes it's not feasible or not realistic to actually go from one point to another point along a straight line. Just like if we go along the Manhattan Street, so we have to go along the road instead of the geometric geometric distance directly link the 2 points. That means the right route is the one we can go. So this right route is simply something along the vertical and the horizontal. So that's why Manhattan distance is introduced. Essentially, that means that we actually look at the separation on each component, on each particular, for instance, the separation of one gene and then separation of another gene, because sometimes the combination of the gene may or may not make sense. In that case, we may simply go individual. So for instance, the gene expression level of one gene in one sample and the same gene in another sample. So each gene corresponds to a particular dimension, right? Maybe along the x dimension. So this is how a Manhattan distance makes sense. So that means for some genetic studies, it makes sense to look at the individual genes, but it may not make sense to simply combine the genes because after all, they have different functions. They may not actually have this simple geometric combination type of behavior. So this is one particular issue we have to be concerned. There's another issue here. When we talk about the similarity between data, sometimes we not only consider the data between 2 samples, we may have to consider the data between 2 sets, 2 groups of samples. Just like this figure shows, we have one group of yellow color, another group of blue color, right? So that's this problem. How do we define the distance between these 2 groups of samples? You may define it simply by the 2 samples that are closest to each other. So that's one definition. But of course, you may define the separation of the 2 groups of samples using the 2 samples that are furthest apart, right? So that's another type of definition. Of course, some people say that the closest separated samples may be may not really represent the whole group. And also the furthest away samples may also may also not represent the whole group. Maybe it makes sense to look at the central points, the sample that is in the middle, in the average position of each group. So that means that we have a third definition of the using the average position of the two group samples to define the distance or the separation or the similarity level between two groups of data. So this way, we actually introduced three types of uh, group distances. The first on the left-hand side, it is called a single link or single linkage, meaning that the shortest distance between the two group of samples is used for defining the separation of the two groups. And then you have this complete linkage in the middle. So in this way, we know that no matter which sample you choose for each of the groups, and the separation is always smaller than the complete the separation defined by the complete linkage. So the distance between the two samples that are furthest apart from each other. Of course, you have this average linkage. So when you use any clustering software, so normally they provide you with the parameters. One of the parameters is this linkage. So you have to choose either single or complete or average. So you may imagine average seems to make more sense, but I can tell you in many cases, sometimes complete linkage works much better than average linkage. Sometimes a single linkage may be better. So this is a parameter that you have to try to get so based on the final outcome, you may decide to choose a particular linkage. So another parameter is of course the definition of the distance. So far we talked about our common sense normal definition of distance, Euclidean distance. We also mentioned about Manhattan distance. There are also some other distances. Okay, so a final comment on the linkage. So the single linkage tend to generate a cluster of long chains. A long chain means that some of its members can be further away from the other side, from the other group. So this is understandable because you deem two clusters to be close to each other based only on the closest samples. So you don't care about the some members being too wandering too far away, right? So that is the cluster you normally get, the long chain type of cluster. If you use complete linkage, you normally get these so-called clumps, so aggregated, tightly aggregated clusters. This is also understandable because if you select you define two clusters to be close to each other, the furthest distance actually sets the limit of the group members. No members can go beyond that distance, otherwise it's disqualified as being a cluster that are close to each other. So that's why it normally generates a clumps type of clusters. Average distance is between these two types. So it is up to your preference based on the problem you actually study, and then you may try these three type of clusters. Okay. Now we come back to the definition of distance. So we are looking, we look at a third definition of distance called Pearson correlation. Normally in some clustering studies, particularly in gene expression studies, Pearson correlation is more frequently used than Euclidean distance, than Manhattan distance. So in this, so Pearson correlation was introduced by mathematician Pearson, named Pearson, obviously, and based on some statistical study of the data. So we just give you the mathematical formula, mathematical algorithm below, without actually going through how it is derived. So you just in the figure above, it shows the type of the shape of the correlation based on the value of this Pearson correlation distance, Pearson correlation value. Okay, so Pearson correlation, if Pearson correlation equals to 1, it means a perfect correlation. If the Pearson correlation equals to 0, it means no correlation at all. If it goes negative towards -1, it is the anti-correlation, still correlation. So the formula here below is actually 1 minus Pearson correlation. 1 minus Pearson correlation is really linked to distance, like a Euclidean distance, like a Manhattan distance. So when you use Pearson correlation, remember that you have to use 1 minus the correlation in order to get the distance in the matter. So the so another point is that in Euclidean distance or Manhattan distance, if a distance a zero distance means they are identical. So in Pearson correlation, of course, the zero distance means the Pearson correlation value equals to 1, right? So then the distance is equal to 0. 1 minus 1 is equal to 0. But when Pearson correlation equals to 1, the two data doesn't have to be exactly the same. It could be that they are simply parallel. They actually change. They vary along with the parameters parallelly, and the Pearson correlation is still equal to 1. So this feature is very good. We know that, for instance, we look at we consider an example. I think most of you should be more familiar with. We look at genes in cancer patients. We want to identify which genes are associated with cancer. Normally, we look at those genes that are elevated, right, highly expressed in cancer with respect to the normal person. In the healthy person, we want to look at those genes that they are normally expressed in normal person, healthy persons, but overexpressed, elevated in cancer patients, right? But those genes, they overexpressed genes, they may not have exactly the same overexpressed value, but they have very similar trend. That means they all get elevated in cancer patients. So we certainly we want to, so essentially that means that they, even though they don't have the same exactly the same expression values, but they go synchronically in with respect to the cancer status. So the more severe the cancer, maybe the more overexpressed those genes are. If this is the situation, we know these genes may be correlated, may be linked, or associated with cancers. So regardless whether they have exactly the same value or not. So Pearson correlation actually provides you with this power to analyze those synchronically varied genes without being this a tight requirement of the requirement of the genes that have the same expression level. Actually, this is we don't really require that. So in that case, certainly we know that Pearson correlation is a better choice for the analysis of gene expressions. Here are examples of the different Pearson correlation values. So you can see as the data distribution changes, so the actually the linear function defined by Pearson correlation also changes. You can see it also changes along with the data type. Okay. Apart from Pearson correlation, if you look into the source codes of different AI algorithms, particularly deep learning algorithms, you'll notice that in many cases, they also need to compute the similarity of the data, compute the correlation. Quite often, they actually use so-called cosine correlation. So here is the definition of a cosine. Essentially, it's just a cosine function of the angles between the two vectors of the two data. So essentially, so that's why the name comes in, cosine correlation. So this is actually popularly used in many AI algorithms. So it's not the Euclidean distance, it's not the Manhattan distance, it's not the Pearson correlation, but rather some programmers, they prefer to use cosine correlation, not because they are familiar with cosine function, but rather they tried different correlation functions as well as different definition of the group of distance, the different linkages. In the end, they arrive at a certain, they found that a certain correlation function, certain distance function works better than others. So that's actually the heart of AI. So you need to try different parameters, different settings. So patiently, you can't pre-assume one is always superior than the rest. So the suggestion is that you need to be open-minded regarding to the choice of the parameters. So that's the situation. Now we look at a specific example about how a hierarchical clustering works, how we actually develop the hierarchical clustering algorithm. So that's the first unsupervised learning method we talk about. So now we have to, so we already defined the distance or the correlation function. We already defined how we measure the distance between two groups of data. Now we need to talk about the task of unsupervised learning. Remember that in unsupervised learning, we don't have any knowledge about the data. So now we simply hand the data to AI. We let the AI to come out with some characters, patterns of the data. So what a pattern AI can look into. So essentially, we know that in nature, something that correlated to each other tends to be closer to each other. Like a city's infrastructure, it needs to serve the population in the city, so it must have the transportation system, a public transportation system, food, the supply chains, electricity lines, and so on and so forth, right? The communication, all this has to be built in close to each other. So this is a general feature. So one important pattern is that if there are some pattern, then those that tend to be associated with each other tend to be close to each other. So the main task of any unsupervised learning AI is to find which samples are close to each other and deem those samples to be within a one group, within the same group. So essentially, a unsupervised AI tries to divide the data into different subgroups and then define the relationship among the groups. A relationship being that some groups may be close by. For instance, you have a family of genes. In each family, those genes have similar functions, and you have a superfamily of genes. Means that a superfamily contains several family of genes, even though they have different functions, but they have broadly similar functions. Just like you have a family of tyrosine kinase, and you also have a superfamily of kinase that not only contain tyrosine kinase, but also containing other type of kinases, right? So here is the illustration of how we can develop a hierarchical clustering to separate samples into groups. Based on this figure, what we can see that because we can see it, so of course we can see that maybe these three should be in one group, or if you define it more broadly, these four should be in a group, maybe this should be in group, and so on and so forth. So for a hierarchical clustering, it begins with the calculation of pairwise distances. If you do pairwise distances, of course, you can immediately identify which pairs are closest to each other, right? Obviously, it is this one. So in the first step of hierarchical clustering, after you compute the pairwise distances, you have a pairwise distance matrix, you simply pick up the two points that are close to each other, and then you group them together into a single group. So this is the first step. And then, of course, the next closest pair, and then after you group these two groups together, right? And then you need to count this one group as a single sample, combined sample. Of course, now comes to the question: how do you define the distance between group 1 and group 2, or group 1 with the sample below? Again, so this is how a complete linkage and the average linkage, those linkages matter comes into play. You have to select a linkage before you can define the distance. Suppose we solve this linkage problem, we choose a particular linkage, and then we can compute the distance between we recompute the pairwise distance. Regarding that, group 1 becomes a single member now. So you compute obviously we notice that the closest distance must be between group 1 and this sample. So this way we have the essentially the third group. So after this, we continue to compute so after this, we of course actually combine all these three samples into one sample, and then we continue to do the pairwise computation until we actually unify all the samples together. So let me recall the process. So somehow I cannot see. So essentially, we group something together in the first step. For instance, we can assign these 2 points as the 7th and the 6th points, x6, x7, and then we can actually use a so-called tree type of graph to illustrate the series of combination process that we combine the sample, the data together. The first is X6 and X7 in combination to form a single new member. And then the next is this one, X1, X3. And also we can define the height of the combination, right? The height up to the combination point to be associated with the distance between the two samples. That means the more distance, the longer the height. So this way, so remember that in this tree is called a dendrogram tree. In this dendrogram tree, actually, the height corresponds to the distance of the pairs. So this is something you need to remember. So this way, we can actually keep combining the samples until all the things are unified into the single group. So this tree is called the hierarchical tree of dendrogram. So this hierarchical tree tells us about the subgroups of the whole data. So remember that the height corresponds to the distance. So now it is up to you to define which distance you deem to be the border for one group that goes into the outside of the group. For instance, we deem the distance to be at this level. So we simply draw a horizontal line here, and we can count that there's one vertical line, two vertical lines, 3, 4, 5, 6. There are 6 vertical lines that actually cross with our horizontal line. So those 6 lines, you can go above, go above to find each line actually contain one or more members. So those members actually are the members of each group. We have 6 groups if we define the group distance at this particular situation. So here we can sense that the hierarchical clustering method is a method that we keep combining closest distance pairs to unify them into a single group. So that's the first procedure. The second procedure is that we define a maximum separation of one group among the members of each group, beyond which it no longer belongs to this group. By that distance, we can draw a horizontal line to actually generate multiple groups. Each group we know that is with one or more members that we know that they are not their separations are not go beyond a certain distance we define. So that's the how the hierarchical clustering works. Now we look at this animation of the hierarchical clustering process. So it starts from so you can see that it keeps combining the samples, and then you see on the right-hand side the dendrogram tree is generated. In this case, dendrogram tree is actually upside down. So you can either way it's okay. And again, the height. Remember that the height corresponds to the real distance. So this way you can see that all the samples are combined into the a single group. So let me. I don't know whether I can pause here. So I cannot. Maybe I should need to pause it here. To cannot. there. Okay, pause. Okay, pause. You have to play until the end, and then I pause it. Okay, I paused it. So it's kind of okay. So from here, we can see that how we define the distance so that we can define the groups, right? So based on this visual, of course, we can see that definitely these three, they must belong to the same group. So now the question is whether we regard these three as a single group, or we deem they actually they are too distant apart, that they can't be considered as the same group. So again, this is up to your own judgment. So in any unsupervised learning, there is one parameter you have to do yourself. AI cannot do. That means how you define the number of groups, or how you define the distance that actually restrict the size of each group. So this is really up to your own judgment. So normally, so that means normally a hierarchical clustering method gives you this dendrogram tree, but it doesn't give you the horizontal line to define how many groups this tree actually tells you. For instance, we can draw a line. Sorry, I think we need to. You can see that we can draw a line here very low, and then each individual member becomes a separated one. Or we can draw a line here. That means that these three actually belong to the same group, right? But the rest actually are individually. Or we could draw from here. That means that these three are deemed to be one group. Then we have three. If we draw here, we can see there are three groups, right? This is the green one is the first group. This is the second group. That means this one. And then the red here, they form one group. Or if you deem they are too close to each other, that all members should be considered as the same group, then you can actually draw the horizontal line here. Means that all of them actually belong to the same group. So this is a parameter you have to do on your own. AI cannot help you in that regard. Okay. So let me continue. Okay, so now the question is how we can do it mathematically to generate the dendrogram, the hierarchical clustering. So we actually start from this. Remember that our starting point is a pairwise distance, right? So we start with this pairwise distance matrix. We have 4 samples, A, B, C, D, and then we calculate the pairwise distance between AB, AC, DC, and BD. So this is the distance matrix. From this matrix, we can see that the shortest distance is this one between A and D. So we combine A and D together. The stocks. When you do the dendrogram, remember that so the samples here doesn't have to be aligned in the original position. You can actually switch the position. If 2 samples they combine into one single sample, you need to move the 2 samples to the neighbor by each other. So that's the process you have to do. So the height, remember the height here corresponds to the value 2 here. So here we combine the A and the D into a single group. So afterwards, A and D is considered as a single point, and then you need to recalculate the distance matrix. We no longer have individual A and D, we rather have a combined AD. So that's a single point. So this is the new distance matrix between the AD group B and C. So we see that it is the AD and the C that has the closest distance. And so we combine AD and C together. Sorry, I think. Again, the height corresponds to the distance. So this way we generated the next step of the hierarchical tree. Remember that in our case, we actually choose a single linkage. That means the shortest distance between the two groups of data. So the shortest distance. Now A, D, and C, they actually are combined into single point. So we only have two points, right? So there's only one distance, and you simply combine them together to form the final cluster. So this is how a hierarchical cluster means. You can simply use the distance matrix, but remember that you have to update the distance matrix after you combine the group members together. So once you have this hierarchical tree, remember that when we do the hierarchical tree, we actually for those the data in combination, we move them to each other in neighbors, right? So that's why you can see that the position of the genes actually changes along with the hierarchical the process until you finally generate the process. So on the right-hand side are the illustratively real values of those genes. You can see that after the hierarchical clustering, for instance, gene 1 and gene 2, they are grouped into this subgroup. You can see that roughly the expression pattern are similar to each other, at least similar than the rest. You can see gene 3 and gene 8, they also have a similar pattern. So this is the power of a hierarchical clustering that can group data that are of similar patterns. Remember that they are similar patterns, but they doesn't necessarily have exactly very close values as long as the pattern is parallel. They can be actually clustered in the same groups. So normally hierarchical clustering tells you that the data, which data actually are close to each other based on this dendrogram tree. But sometimes we also want to know, for instance, when we do gene expression analysis, we also want to know what are really the expression value of the gene. Of course, we could use this curve to show that. But if we have too many genes, for instance, dozens or even more genes, if we have gene expression value that are not really so obvious, maybe changes so in a complicated way. Sometimes they have smaller variations. So this way, it's better to use this so-called heat map, this color schemes. So that means that we have remember that when we talk about the gene expression, we have normally we are interested with certain genes that are elevated, overexpressed. Certain genes, they are downregulated, right? They are become more silent. So if we can use colors to actually display this kind of gene expression level, it's much better visually than looking into the curve of the gene expression. So that's why people introduce this heat map along with hierarchical dendrogram trees to display the clustering results. So the red value, the bright red, means the highly expressed or larger values, and then it goes darker until it becomes green and bright green. The bright green means the lowly expressed, downregulated gene, or sometimes, depending on the data, maybe the negative value of the data. So in between, you have this dark black region that corresponds to the normal expression genes, genes that are neither overexpressed, downregulated. They are just normal. For the normal genes, they are assigned with this dark black values. So this is the heat map. So we talk about the heat map. We talk about the dendrogram, the hierarchical tree, and then we look at this particular case, right? So we know that normally when we do hierarchical clustering, we have the samples. Each sample may contain. Actually, we measure multiple genes for each sample. Like for instance, we do the measurement of do this blood test, the blood genetic test. We may measure the expression of multiple genes, many genes. So obviously we have this heat map to reflect the multiple genes, but also for each horizontal line, it corresponds to a particular sample, or else you could consider each vertical line as a sample and each horizontal line as a particular gene. Either way, it is okay. So anyway, we have two situations here. So one situation is that we may group genes together, genes with similar behavior, or we may group sample together, patients with similar disease, similar patterns. So you can do two types of hierarchical clustering. One on the sample side. So here we can define each vertical line as a sample. And you can also, so that means that we can switch the vertical lines so that the lines with the same behavior, they actually get close to each other. We could also simultaneously cluster genes, the horizontal lines, meaning that those horizontal lines, they have the similar behavior. They are positioned closer to each other. So normally a dendrogram heat map essentially gives you both hierarchical clustering results, clustering in patients, in samples, clustering in genes. So that's the normal situation. So far, we talked about this hierarchical tree. I wonder if you notice this problem, so-called leaf ordering problem. So let's look at the examples here. So we have one cluster, right? One subgroup of samples or genes, depending on how you define that. So we have another group here. We notice that this problem. For instance, why do we define, for instance, in this subgroup, we have two samples, right? How do we define which sample on top, which on bottom? For this is the same thing, which on top, which on bottom. Also the same thing here. And depending on this kind of, so this is so-called leaf ordering. That means we can order the leaf. So we regard each color stripe as as a kind of leaf, right? So we can actually reorder the leaf in many different ways by simply turn over the order, right? Turn over the order. So and we know that visually, if you do this switch, we know that the pattern can look very different. For instance, we look at this one. even though we know superficially there's no, it's meaningless to actually to prefer this particular sample or gene to be on the lower part instead of upper part. But we know that if we position at the lower part, it actually visually looks better than otherwise because we have a red that match the red below, right? So it makes it even better. It's a similar thing here. If we do the switch and we can have the essentially some green that actually goes below, and that it becomes closer to the one here, right? Because the visual so-called visual effect. So this is actually a big problem. So that's why sometimes if you use different software, even though you choose the same parameters, like I mentioned, the linkage, distance definition, cutoff, and so on and so forth, you still can have quite a different heat maps. That may be because of the leaf ordering problems, because different software, they have different way to actually define the ordering. So this is really again, there's no good solution here. You just have to try different algorithms. Okay, so much so about hierarchical clustering. Now we look at a second method called k-means method. Okay. In this case, on the left-hand side is the data distribution. So based on the density of data, it's seemingly that we can either consider this as a group, this as a group, this as a group, or maybe this as a group, the other as a group, or maybe this as a group, the other as a group, right? So we can see that based on the density, there must be some focal points or the smaller region, the core region of the data that seems to steer us from one option to the other option in terms of the definition of the group of data. So that's the visually we see that if so normally when we human consider the separation of data, normally we look at the density, the core regions, because we know that the core regions, they must be close to each other. And then we consider the vicinity of the core regions. Maybe it should be close to this side or to the other side, right? So this group of data based on core regions gives us an idea about how we introduce this so-called k-means approach. So essentially, we need to first define the focal points, and then we judge each sample to see which focal point it must go into. And so this is called the k-means algorithm. So we look at the simplest example of one-dimensional data. So the data is given on the top. So there are 4 data points on the one-dimensional on this straight line. So if we define, we want to group data together. Obviously, we know these two, they close to each other. We simply group them together, right? Now the question are these two, how we define them to be same group or different groups. So if we deem them to be too further apart, then so this is the good definition of the so-called central point. So we define the central point here for these 2 samples, and then define the central point for these 2 individual samples because we deem them to be further apart. But if we deem them to be close to each other, then the focal point must be here. Of course, there's this question. How do we cannot visualize the data at all. How do we know how many, so that's the first question. How do we know how many central points we should introduce? So that's the first question. The second question is, how do we know where we should position those central points before we can apply the k-means, right? Remember that k-means rely on the position of the central points and as well as the fixed number of central points. So I can tell you why it is called the k-means. K is an integer value define the number of central points or define the number of clusters. Okay. So how do we know the k? So this is similar to the situation of kn. There's no answer. K is a parameter you have to try on your own. So that's the downside of any unsupervised learning method until the transformer comes in. Before transformer comes in, normally you have to define, particularly in machine learning method, you have to define the number of clusters on your own. Okay. So first we define the distance. So the distance in k-means, surprisingly, most people simply choose Euclidean distance. Euclidean seems to perform better in many situations, but again, there's no, it's not always the case. So sometimes you may try other means. And also, this is a formula to calculate the central point. It is simply the average position of the average value of the members, memberships. Okay. Of course, here we have not solved how we define the number, how we define the initial position. Okay. Now let's look at the real situation. So here is an example of the data distribution. Okay. From this data, we can see that definitely this forms a group, right? So they close to each other. This forms group, this forms group. So ideally, if the central point, so there must be 3 central points. If the central points are located here, we know that we can get a right answer more or less closely. So suppose that these 3 points are chosen as the central points, and then we simply calculate the distance. We choose any point to calculate distance to these 3 points. And then we assign the group membership based on the distance closest to the central points. Obviously, these actually are grouped with this. These actually are grouped with this central point. So after the grouping, we recalculate the central points. We notice that the central point should move because the average. So the average is not really not necessarily our initial choice. So the average, for instance, the average of these 3 points obviously is not here, but rather it's in different position. So you can do the same thing. Okay. And this way we can arrive a final group with the perfect central points. So illustratively, I think you can understand that. But the question, now the question is, what if our central point was chosen incorrectly? Can we still arrive at a good clustering, a good grouping by this k-means clustering? So here we look at an example. So we have, of course, we randomly divide the sample into 2 groups. The green, blue one, and also the red one. So this is a rough border. And we randomly selected this as the central point, and this as the central point. Obviously, this central point is incorrect because it's closer to the other side, but it doesn't matter. We simply calculated it, and also we know some members may be incorrectly assigned, so we recalculated the distance, and you can see the membership should change, and also the central point should shift from the original position here towards the true center points. So this way we can see that the machine learning process can correct the wrong choice of central point by itself gradually. But we ask another serious problem. What if we totally get the central points wrong? So this is second example here, another example here. So in this example, we see that these points, they must belong to the same group. Maybe these two, and also these form the third group, right? Suppose that we choose the central points as we choose the number of central points correctly, three group, and we choose the position totally wrong. For instance, we choose the position of the two clusters here, and then the third one is here. After all, we cannot really see the data, right? So this is a more realistic case. We got totally wrong, got the central points totally wrong. So we calculate the distance of the points to these three central points, and then assign group membership based on the closest distance to the central point. So this is the first round. We can see that only this member goes to this point, all the data go this one, and also this actually goes to this one. And then based on this, we recalculate the new central points. So the new central points becomes here. And then we calculate the membership based on the new central points. So we can see that some data actually are rearranged or regrouped, reassigned to the three central points. So after the reassignment, we again recalculate the distance, the new central points. You'll notice that actually the process corrects itself and then finally arrive at the more or less the true central points. So this is the beauty of a machine learning method. So you normally after so many tries, it will finally settle down at the true central point. But there's a catch here, provided you make the right choice of the number k. But unfortunately, that number k is up to yourself. Okay. So here is a animation of the central points. You can see that by choosing the central points initially, and if it is in the wrong position, after several rounds, it will find its true position. So another issue here, how do we define? Remember that we have to define the number k by our own, right? So how do we define the k? How do we know whether our choice of k is correct or not? So you can do this by a measurement that measure the intergroup distance, and also intra-cluster, intra-group distance, and then you can look at the ratio of those two. If the intergroup distance is not much larger than the intra-group distance, you mean that the intra-groups, they may be too close to each other. They may need to be combined each other. So that means maybe the choice of k is too large. You may need to reduce the k. If otherwise the intergroup distance is much larger than the intragroup, you may consider maybe the number of k is too large. You may need to too small. You may need to increase the number of k. So these are the guidelines. Okay. Finally, we look at a third method called the fuzzy C-means. Essentially, it is a K-means, a generalization of a K-means. So fuzzy C-means actually can be divided into two groups. The first is the C-means, which is essentially the same as K-means. It's because it's crisp. Because fuzzy means it is not really absolute, really black and white, right? There's some kind of fuzziness in that. Fuzziness being about the membership. Crisp means that the membership is clear, right? So that's why people instead use this C means, which represents crisp means instead of K means. So C means the crisp. So actually, our previous the discussion about the K means essentially just the crisp. So the membership is clear, either belong to this group or the other group, but there's no between. But we know there's a situation that sometimes a particular gene could belong to two families, right? So the membership may not be that clear cut. We should sometimes allow a sample to be, particularly those sample on the borderline, right, to be have this kind of dual type of membership. So how do we with this dual type of memberships? So here we have to introduce some mathematics. So remember that so we look at the formula here. So remember that. So we have a central point C. So I means the index of a cluster. So we have a C number of clusters. Remember, we are talking about the C means. C represents the K here. So that is so we have a C number of clusters. Each cluster we have a central point. We have the membership. A member have a distance to the central point. So we have a combined distance. This combined distance is called a cost function. A J function is called a cost function. Cost function is a very, very important concept in deep learning, in some machine learning. So that's why here we consciously introduce the cost function here instead of simply talking the so-called the obvious fact, right? So the obvious fact is that our members has a certain distance to the central point. We want the members to be closer to this central point instead of the other central point. So that's why we have a distance measure to decide whether this particular sample belong to this particular point. So this is called the individual distance, right? And then the cost function is simply the sum of the distance functions. Okay. Now we consider a situation in which the member here doesn't have to totally belong to a particular I. It can be either belong to this I or the other I. In this way, we have to introduce an additional parameters. So illustratively, that means that we have a situation that a sample in the borderline that can have certain percentage belong to one group, a certain percentage belong to the other group. So that's the illustration figures. Okay. Okay, so we change this formula slightly. Remember that we have this distance, right? And then we introduce an additional parameter called the weight, essentially our component, fuzziness component. So in our original formula, this is the distance. Original formula, this actually is equal to 1, absolute value. Here, we allow it to be a fraction of 1, in particular I, maybe fraction of 1 in another value. So this is the way. So this way we introduce an additional parameters. So we have additional sum operations. So this is the fuzzy sequence. So actually, this type of cost function relying on certain parameters actually is frequently utilized in deep learning algorithms. So here we introduce the same concept with this dual membership or multiple membership of the samples. We can have a complex algorithm to actually group data together to form the data. So much so about today's lecture. So maybe it's time for questions.
Yeah, thank you very much for presenting your Right. With your permission, do we have any question here from the in-person audience? Any question? Okay, so there is one question in the chat box by Mr. Aspandyar. He's asking, what are some matrices we can use to choose K in K? K means clustering, if we are unable to visualize the data.
For k-means, normally we don't visualize the k-means data. So actually, when we consider visualization, normally we choose hierarchical clustering because when we do visualization, that normally means that the number of samples are not that large. If there are too large number of samples, whatever visualization is just simply a mess. So if you have smaller, for instance, if we just zoom in on a few dozen genes, so why not just the hierarchical clustering? It gives you a better visualization because it has a heat map. Of course, there are some other type of clustering method, but certainly not k-means. K-means are normally used for data with a large number of samples, because if you have too high number of samples, you cannot use hierarchical clustering algorithm-wise and also visualization-wise. So for instance, if you do a transcriptome of a certain patients, right? So the transcriptome normally has the gene present value for so either 20,000 genes or less than 20,000, maybe 15,000 genes. You can't display a heat map for 20,000 genes. It's going to be a total mess. So you need to focus. So that's my suggestion. That's the reason why people actually did not actually design a visualization for k-means. But of course, if you really want to see the distribution of the data, you can explore. There are some dimensionality reduction algorithms to actually display the data distribution, either data generated by k-means, because by k-means it actually gives you the label. With the label, you can actually color the data to generate the kind of data. So normally k-means are associated with this dimensionality reduction algorithm. The simpler one, the simplest one, and also most most traditional one is the PCA. So we are going to devote a lecture to talk about the dimensionality reduction algorithm, but not today. Okay. Okay. Because of the time, because we have to give the Dr. Wang time to do the practicals. Okay. So any other questions?
No, this is the only question. And
Okay.
when Dr. Wang, when does he want to give the practical session?
Okay. I quit the sharing and I let Dr. Wang to give you the. Quit the sharing. Eh? Not, how can I quit the sharing? I already quit the sharing, right? Dr. Wang, can you hear us, Dr. Wang?
Okay, so Professor Chen.
I contacted him to see.
Yes, I'm going to add them. So I just want to explain one thing. That is that before your lecture, as announced yesterday, before your lecture, all our participants who were present here in person and also online, they have already downloaded Anaconda suite and have done the pre-installation. So you want to give time to Dr. Wang for only today or for tomorrow as well? Like how do you want to, because is it only one lecture or you want to give a practical session time to Dr. Wang? Dr. Chen?
I'm contacting Dr. Wang, so I don't know why he has not. Yeah, he responded.
No,
He doesn't have the right. Can you give him the right?
I'm going to give him the right, sir. I'm
Okay.
asking how much time does he need?
I don't know.
Because
Ask him.
I actually told him that we will be doing practical session today at 12:45 before your lecture. So we have done that with the help
You
of
you have already completed the practicals before the lecture. You mean? Oh.
Yes, the installation process, the pre-installation process.
Oh, you already go through, but I thought we should practice k nearest neighbor, not k means, because
Okay.
we practice. Actually, so if you practice k means, it means you practice it before the lecture, right? So that's why I asked Dr. Wang to actually practice KN. I think he misunderstood that, so I apologize for that.
No,
So if it's already
I-
done, that's fine. If you found it, so I think we can complete the better. Anyway, we learned the K-means today, even though it is after the practical. But anyway, a practice means that you have to practice yourself after the class, not on the class, because the time is kind of short, right?
Yes, so I'm just asking for tomorrow and the next lectures that you have. Do you have practical sessions in all those lectures?
No, every 3 lectures. That means the next one is a Friday.
Okay, right. So
Today is Tuesday, right? Tuesday,
yes,
and then Wednesday, Thursday, no practicals, and then the Friday,
okay,
Tuesday
right.
and Friday, yeah.
Okay, so
Because
you
we
want
wanted the students to try it, so in the 2 days, the following 2 days, because you need to get some data set to run it. It's better to practice like a real test. You get a data set, you use the data set to. So that's why I think in the beginning we should try k-mean, no, sorry, the KN, the classification. And then later on, we can try more sophisticated the machine learning,
okay, so
deep
sir,
learning,
so
random forest. I think the next one is a random forest, but we have to try KN first before we can try random forest.
okay, sir. So on Friday, we will do a practical session, and
Yes.
that will be after your lecture, right?
Yes.
Okay. So
It can be before the lecture, as
Okay.
long as the practical the content has already been taught before the practical. That's fine, no problem.
Okay, right.
It
So
can be before the lecture. It's better to be before the lecture, actually.
okay, right. So
So
I will talk to Dr. Bag, and for today, I think it's okay. Thank you very much for your time.
yes.
And we will do
Okay.
the practical session tomorrow,
Okay, thank you.
on
Okay.
Friday. Thank you very much. See you
Okay.
tomorrow.